Aloha and welcome to our next video. This is 6.4 Meiosis, the life cycle of sex cells. So it'll be a little bit of a different one. Once again, this is just a simple quick review of the materials that we'll cover in our lesson. Okay, for this video, hopefully we will be able to compare the number of chromosomes between somatic and gametes. We'll also describe the stages of meiosis, and then finally we'll compare meiosis and mitosis for you. So part one, let's look at the numbers of chromosomes in somatic cells and gametes. Okay, so here we have a nice little chart that shows us we have two different types of cells in the body. We have our somatic cells, which are our body cells. This is just about everything in your entire body, except for the gametes. The gametes are going to be the sex cells. In females, this will be the eggs, and males, this will be the sperms. In somatic cells, our cells are diploid, which means they are 2N. That means that they have two sets of chromosomes. Okay, and remember, if you can recall back, you got one from your mother and one from your father, so that's how we end up with two sets. So for every trait, there's two different genes that'll set it up for us. Okay, so in humans, there are 46 chromosomes in the somatic cell. Of those, there's 23 homologous pairs. So 23 of them are pairs of the same chromosome. You just got one from mom and one from dad. In the gametes, it's a little different. We have what we call a haploid condition. And that's where we have only one chromosome. So you no longer have the pairs. We have to split those pairs up. So in us humans, it's only 23 chromosomes. So we kind of got to remember that our body cells are diploid. They have two sets of chromosomes, one from mom and one from dad. And then our sex cells only have one. And the reason for that is you're only going to want to give one away. Otherwise, we would double every time and we'd have way too much DNA to do anything. Okay, so now let's take a look at meiosis and see how the cells get from the diploid to the haploid condition. Okay, meiosis is only found in the gametes, so it's only found when we're making eggs and when we're making sperm. It's used to make a haploid cells for this reproduction, and it results in a cell that only has 23 chromosomes. So instead of having the 46 chromosomes, we're going to cut it down into half, into 23. So here we can take a look at meiosis. We can see it looks an awful lot like mitosis. Okay, we're going to line up, the phases look about the same, so we'll kind of go through there and we'll kind of take a picture of them real quickly. First up, if we look over here into our prophase, okay, so this first one over here, we have our original cell. Our chromosomes are in there, they're thickening up, our nuclear membrane's starting to disappear, the chromosomes are going to be paired up, okay, and then when we get over here, notice, remember now, there's 46 of these chromosomes, so they make pairs. So when we get into metaphase, it lines up a little bit differently. If you'll notice, we see our chromosomes that are lining up this way. And then what that does is it pairs up these homologous pairs. So they're all together, they all match up like that. Then during anaphase, we break these homologous pairs apart. Okay, so that's what I want you guys to see, this difference. Okay, so these two here make a pair, and then notice during anaphase, they separate. So when we get into telophase 1, what we're talking about is we've split apart those homologous pairs. Now, in doing so, we might separate out mother from father DNA. Okay, so you might have, this one here might be from your mother, this one here might be from your father. So that's why your siblings, your brothers and sisters, look a lot like you, but they're not exactly like you, because it's actually, we get a little variation this way, and that's really important for the continuation of life. Okay? Also, when we're talking about this, we have this crossing over that sometimes occurs. So if you look at the picture at the bottom, what we have is, let's just call this one over here your mother chromosome, and this one's your father chromosome. So while these guys are lining up in the middle here, every once in a while they kind of cross over, like you can kind of see occurring right here. And then what that'll do is, if the DNA lines up properly, it'll actually start switching pieces of DNA. Okay, so you might not have an entire mother DNA chromosome. It might have a little bit of father that happened with crossing over. And this again is important because it introduces in variation, and that's what we like to have. That's what makes life successful. Okay, so we got through the first part of meiosis, and we took our cell, and remember we split it into two of these. Okay, so here's one of them, here's the other one at the end of meiosis 1. During meiosis 2, we get right back into the prophase, gets there, metaphase, everything's going to line up again, and this is actually showing us a little more metaphase than it is prophase, but that's okay, we understand the process. Okay, during metaphase and anaphase, what's going to happen is now we're going to separate out the sister chromatids from these chromosomes. Okay, so you can see the centromere in the middle here, and our spindle would attach to that, and we can see how this one here is now being split up into that there. Okay, so we're splitting out the chromatids. Okay, so first we had the chromosomes, the big ones, and now we're splitting out the sister chromatids. And then that's going to take us through telophase and division. And because we're going through and we're not duplicating this DNA, if we stopped right here, okay, then we could go back into interphase, we could make copies of our DNA and we would have an additional set. But we're skipping that part this time and we're splitting it again. And that's how we get from the diploid to the haploid condition. Okay, and that's really important for us. Okay, so let's review the steps real quickly. In prophase 1, our sister chromatids shrink up, they thicken up, the spindle fibers attach, and the synapses and homologous pairs together, and they form the tetrad also occurs. So that just means that all of these pairs, these homologous pairs of chromosomes, are going to get together. They call it a tetrad because there's 4 chromatids, okay, 2 on each of the chromosomes. So when those pairs split, we split it up into 2 and 2. Okay, metaphase, everything's going to line up in the middle of that. Anaphase, we start splitting the homologous pairs. Okay, and that's how we separate it out from being the 46 to the 23. The nuclei and the cytoplasm divide in half during telophase 1. Then, without going into interphase and copying the DNA again, we start the whole process all over. And that's what allows us to go from a full set of 46 down to a set of 23. Okay, and that's the part that we need to understand that's going on. So meiosis 1 separates out the sister chromosomes. Meiosis 2 breaks apart the chromatids. And then we end up with our 4 cells that have the haploid condition. Okay, so let's look at meiosis and mitosis real quickly. We've talked about it the whole time, so it should be kind of a quick review for you. Okay, similarities, like mitosis and meiosis, it's a cycle of division. So basically we're dividing the cell, okay, and this is what we need for supporting life. Nuclear division involves the same process. So the same actions are occurring each time. We line up the chromosomes in the middle, we split them apart, drag them to the poles, and then we separate it that way. The difference is mitosis is only one division. Okay, it creates 2 somatic cells. In meiosis, it has 2 divisions. And it creates these 4 gamete cells. Okay, in meiosis 1, we separate the homologous chromosomes, and then in meiosis 2, the sister chromatids are split apart. Okay, so that's kind of what I want you to understand, that we have that second division, and that second division is what takes us to the haploid condition. Okay, that's all I have for you. If you have any further questions, get in touch with me. Otherwise, feel free to check out the unit lessons, and those will help you as well. Thanks for watching.